mentorship. Our two students for the last year were Zhang and Sheena Caligara, who are both students at Sinai. So we'll start with Jack, and Jack, I'll give you a quick introduction. So Jack was an undergraduate student at the University of Rochester, majoring in chemistry and psychology before coming to Mount Sinai. He completed a master's in science and biomedical sciences and is finishing his MD this year. He is going into neurosurgery, so he's also doing a sub-I with us this month. And he did research he spent 2 years working with Dr. Hadjipanias before he went to Pitt, and then spent a scholarly year with Dr. Zoe. So he'll be talking to us about his work in Dr. Zoe's lab in the last year.
Thanks for the introduction, Dr. Morgenstern. Can everybody see my screen and hear me? Excellent.
Yes.
Let me switch my pointer. Okay. Good morning, everyone. Thanks for being here today, and a sincere thanks to the department for the opportunity to participate in the sub-I and present this talk. Today, my talk will be on a main project of my scholarly year in the lab of Dr. Jenny Zhou, where I studied the role of a class of proteins called plexins in GBM. So GBM, as we all know, GBM is the most common malignant primary brain tumor with a very dismal diagnosis. And the current STUPE protocol was established in 2005 and hasn't really majorly changed in the decades since. And unfortunately, neither really have the survival statistics. So just diving right into it, there are various treatment challenges associated with GBM and kind of contribute to it being such a different treatment target. And I'll focus mainly on the three most relevant ones today that are relevant to my subject, namely its heterogeneity, recurrence, and also its aggressive invasiveness. So the first off, GBM is extremely heterogeneous in itself. So a lot of recent work has been done to understand the different cellular states within the umbrella of GBM. One example is the famous 4 Neftal states that is probably at least 6 years old now, each of which are associated with their own transcriptional programs, driver mutations, and also cellular behaviors. But beyond just the tumor cells themselves, we actually have a very interesting phenomenon of the tumor microenvironment, or TME, which plays a very important role in conferring GBM treatment resistance. GBM doesn't exist in just an isolation as a ball of tumors. It thrives in a highly dynamic, complex, and very poorly accessible microenvironment. So a GBM tumor mass actually contains many non-tumor cells as well. In fact, about half of all the cells present within a GBM lesion may actually be comprised of these TAMs, or tumor-associated myeloid cells, which encompass microglia and also macrophages. And these TAMs are really their own talk entirely. They're fascinating. But in short, they interact with the tumor cells in a lot of different complex ways, and they play roles in mediating immunosuppression. GBM is famously very immune cold, treatment resistance, and even helping the cells invade deeper. And along with these TAMs, there are also various other cells and factors present in the TME, which are all then compartmentalized into these various identifiable niches, some of which are located here, and each of these are very active research areas of research, like hypoxia, angiogenesis, and et cetera. So a second factor of GBM that makes it so hard to treat is its high propensity to recur. A large role of that is played by these glioma stem cells, which are these small subpopulation of the overall heterogeneous cell mass that are capable of self-renewal and regeneration, and hence often drive the recurrence. And so here's just an intra-op screenshot captured during a fluorescence guided resection. So the GSCs are present in the small rim of margin that's left over from resection if GTR is not achieved, and these ultimately drive regrowth, often very close to the margin that was left. And finally, a third and perhaps GBM's most infamous factor is its aggressive invasiveness. So malignancy, when we say GBM is malignant, it's slightly different from malignancy in other solid tumors. Other cancers tend to metastasize through fluid channels like blood or lymph, but GBM actually migrates through the extracellular spaces of the brain, and it squeezes through these tight spaces within the brain parenchyma itself. And so here are some of the structures that GBM has some propensity towards migrating along, which include solid structures like fiber tracts and blood vessels. And in this sense, GBM cells actually act a lot like neural stem cells during normal development, which is a very interesting concept to think about. And they kind of just, they move from one location to another and migrate in much the same way. So it's a very well-known fact that generally cancers hijack existing mechanisms. So this may not be extremely surprising, but how GBM achieves this is a bit more complicated than it might sound. So in cell biology, there's a concept called durotaxis, where the stiffness of a membrane really influences a cell's behavior. And so GBM cells have been shown to prefer more rigid environments, such as axon fibers and blood vessels that we mentioned earlier. And they have also been shown to migrate towards harder surfaces. So they kind of seek them out. And additionally, they also seem to be more aggressive when they're on harder surfaces, and they actually upregulate extracellular membrane proteins within the tumor microenvironment that they then generate to stiffen it. So they really like to make themselves extremely cozy. And that's all well and good, but GBM cells do eventually, as we know, they do eventually detach from these environments that they create for themselves, and they initiate migration actively into softer brain parenchyma. So this overall question of what drives GBM cells to detether from their established networks and leave to invade soft brain is not very well understood, and the mechanisms are, need a little bit more work to elucidate. And additionally, these spaces are extremely tight. Brain tissue is very densely packed. So as GBM cells are squeezing through these areas, they experience really intense physical forces like compression, drag forces, tension, and of course, intracranial pressure from the skull being a closed environment. And so therefore, maintaining proper membrane tension is really important for the cells to, not explode and actually be able to do what they need to do, which is unfortunately is to invade. But even beyond just mere survival, our lab recently showed that membrane tension actually plays a key role in migration as well, in the actual mechanisms of migration. So membrane tension serves as a key link between the cytoskeletal mechanics of the cell and membrane processes such as endocytosis. And these are really important in actually driving cell movement through space. And of relevance to this talk today, this is a separate project that I'm helping on. But for this project, we're focusing on this class of proteins called plexins, which I'll get into on the next slide. So as tumors tend to commandeer developmental pathways, examining these developmental pathways is a good place for us to start our investigation. And so that's naturally where we began. So guidance molecules overall play a key role in mediating cell migration and patterning during development. I'll give a few examples later on. But overall, chief among these are these very ancient and well-conserved proteins called plexins and their partners, which are semaphorins. And they play key roles in development. And they mediate very large-scale changes in the cytoskeleton and cell adhesion. And even more interesting for us in terms of identifying study targets is that Plexin-D1 is actually upregulated in high-grade gliomas and is also associated with the reduction in patient survival. So today's talk will mainly focus on Plexin-D1, but we can talk about other plexins as well if people are interested. So to wrap up the introduction, I'll delve a little bit deeper into Plexin-D1. So Plexin-D1, in conjunction with its semaphorin, it plays a really important role in both CNS and actually cardiovascular development. So in the CNS, it drives the development of the cortex as well as synapses. And in the cardiovascular system, it's key in actually shaping the evolutionarily conserved anatomy of the aorta and the arterial tree. And we can talk a little bit of how it mediates that as well. It's a very a functional protein. So with that, I think that's enough overview. Moving on to our experiments, just like a brief structural note, I'll talk about the in vitro experiments, which I helped out with a little bit, but mainly laid the groundwork for when I started my scholarly year, which was primarily the animal work and moving forward. So to study Plexin-D1, we generated a lentiviral vector that uses a guide RNA to knock out this aspect of the exon present in the human Plexin-D1 protein. So that effectively knocks it out, and then we transfected it into a patient-derived glioma stem cell line. So these cells were derived from real patient samples. They're called SD3 cells. And here's just a Western blot that I ran showing on the left column, this is control, which is a viral vector directed against GFP. It's not present in the cell, so nothing happened. It was just controlling for the presence of a guide RNA. And here we have our actual plexin knockout. As you can see, the expression is completely ameliorated or completely eliminated, rather. So using that as a starting point, we moved on to our in vitro studies. So next, we compared the viability of these cells. So here is a microscope image of these cells stained with Ki-67, a very popular proliferative marker. And the quantification here is that the proliferation is roughly comparable. And then we kind of recapitulated with a similar assay called a scratch assay, in which we plate them on a plate, and then we essentially use a syringe tip to just make a wound between the cells, and this assay allows us to measure wound healing. And when we ran the statistics, they're comparable. And the point of these two kind of foundational experiments is just to show that deleting Plexin-D1 doesn't cause these ST3 cells to become non-viable, which allows us to move forward in comparing the two. Because if one just didn't grow at all, then it would be a false start for the experiment. So next, we looked at the morphology of these cells in culture. At the top here, you can see that the knockout cells actually form these interesting structures that look like tethers. And here's a blown up image and a quantification of the number of tethers that we observed. And on the bottom here, this is just a structural analysis of these tethers. So we stain for phalloidin here in red, which stains the actin cytoskeleton. And then here in white, we have beta integrin, which is a protein that binds to actin. And tubulin is an additional, it's a microtubule protein. So we all these kind of point to the fact that these are actually these very interesting structures. It suggests there's some argument as to whether they're true nanotubules, but there's these tunneling nanotubes that are very interesting aspects of GBM, where the cells kind of connect to each other, sort of like gap junctions, but more GBM specific. And they basically allow the cancer cells to exchange various things, including mitochondria. And they make each other more resistant against medications and chemotherapy. So here, I forgot to mention in the lower corner, we also did a staining for mitochondria and showed that there are indeed mitochondria present in these structures. The reason why I say it's a little up in the air is because we're not yet sure if they're actually trafficking between each other. It could just be a membrane extension, but that remains to be seen. So as these findings suggest some modifications in membrane tension, which we know is important in cell migration, we studied migration using live recordings of cells moving through these specially designed microchannels. So here's what they look like in real time. And essentially, the cells are allowed to grow in these blue ports, and they kind of move through these very thin channels that have a divot in the middle. Basically mimics these points of confinement that cells have to kind of pass through in the actual brain parenchyma. So this is a confined migration assay. And we have these beautiful videos of these experiments that I unfortunately didn't include in here because it was making my computer just go insane. But I just have this screenshot, and the takeaway here is that the Plexin-D1 deleted cells just don't migrate as well as control cells. So here are some quantifications. Overall, we see that the distance that they move is reduced, but not only that, the distance both forward and backwards moved is also reduced. So these cells are just not very good at moving in any direction. And we also, essentially, if we watch the videos, we basically see that they form these tethers over here, and they are kind of just pulling on each other every time one cell tries to move. They're sort of stuck together. And here's a quantification in the bottom right corner of the tethering that forms. So wrapping up the in vitro arm of the study. So as a summary, these knockout cells seem to grow just as well as the control cells in culture. These knockout seems to lead to reduced cell membrane tension, which manifests in various interesting findings, including tether formation and as well as impaired confined migration. And then finally, since these tethers may potentially be nanotubes, that would be interesting in the aspect of studying how they contribute to treatment resistance between the cells present within a single tumor mass. So with that, I'll move on to the in vivo study. So the study, the role of Plexin in vivo, I performed intracranial transplantations of these human-derived tumor cells into the brains of mice. Briefly, I anesthetized a mouse. I made a small scalp incision, and I drilled a burr hole according to stereotactic coordinates, which is roughly in this location. And then I injected control versus Plexin knockout tumor cells to a certain depth to target the striatum, which is the area that we really like these cells to proliferate in. And the mice were then allowed to recover and then sacrificed 7 weeks later and perfused. And then I harvested the brains from the skulls. I embedded the brains in OTC, and then I cut them in 20 micron sections on this cryostat to generate the final tissue samples that we studied. And this is a representative image of a section. And here it's stained with H&A, which is an antibody that we love in the lab. It's human nuclear antigen. And it's great for us because it stains the nuclei of all human cells. And since these tumors are patient-derived, it stains the tumor cells beautifully, but doesn't stain the surrounding mouse cells. So it allows us to really localize the tumors. And here you can see this enormous tumor bulk that forms along the injection track. And then you also see that the cells kind of migrate across the corpus callosum to the contralateral hemisphere. And then I really like comparing it to and I know that the textbook butterfly glioma is not necessarily what we always see, but I think it's a pretty apt comparison in terms of phenotypic resemblance to the human condition here. And so here's an overview of control and knockout sections together. And we can immediately notice that there's a striking difference in extent of invasion in the controls. So versus knockout. So in the controls, we see them kind of seeding all the way over to the contralateral hemisphere and even down to the contralateral striatum under the corpus callosum. And the KO cells really barely make it past midline. And here's just a quantification of the cells that make it past midline. And we also importantly see that tumor size is not dramatically different, which is in line with our in vitro data, kind of showing that they grow about the same. This means that this migration difference isn't just because the KO cells aren't healthy. They are growing. There's just something different about the way they migrate. So here we're going to look a little bit more closely at the contralateral corpus callosum here. Up here is just like a general map of where every single slide I'll be presenting on is focusing on. So it'll be the same regions for controls and knockouts. So once again, we see a striking difference in the extent of tumor cell invasion to the other side for both control and knockouts. It's really not even close in terms of number of cells making it across. And here are the same sections, but I stain them with MBP, which is a myelin basic protein that labels these white matter tracts really well. So here we're looking right below the contralateral corpus callosum at the striatum. So if we look at this image, these bright green bundles are these fiber tracts that are running, out of the screen at us. And we can also see some of these red tumor cells that are associated with these axon bundles. We can see, as we know, glioblastoma cells like to travel along these secondary structures of Scherer. And when we switch over to the red channel, we can once again see a striking difference in degree of invasion between control and knockout cells. So very few, if any, knockout cells actually make it to the contralateral striatum at all. So now we're looking at invasion here, but in a different region, sort of in the lateral most invading edge of the tumor bulk. So as you can see, the control tumors are infiltrating all the way out to the edge of the tissue in the controls, whereas in the knockout cells, they really barely make it out of the bulk. If you look closely at the bulk as well, you'll see that tumor cells are really bunched together. And this could go back to the loss of membrane tension again, where the cells are just connected to one another and unable to detether from this mass that they formed and the environment that they've created for themselves. And now we'll return to the contralateral callosum, once again in a similar region for both controls and knockouts. So in this case, the tumor cells are staying with an NCAM, which is a neural cell adhesion molecule. It's human NCAM. So it labels the cell bodies of all these human-derived tumor cells. And before we talk about the differences here, I just want to first appreciate that what a tumor cell looks like actually in the midst of confined migration. So we can see that these cells are literally getting squeezed by the tight fibers of the callosum, and they travel under these immense pressures and forces that we talked about. Both their nuclei and actually their cytosol get compressed into this fusiform, almost streamlined shape, which is very interesting. But comparing these two control and knockouts, we actually see that there seems to be a length difference between these. The knockout cells seem to be stretching out even longer than the controls, likely due to a softer, more pliable membrane due to loss of plexin. It also seems like the knockout cells have more of these tethers that we observed in vitro, which we just don't really see in the controls. And here we're looking at the midline of the callosum before the tumor cells really make it to the other side. So once again, same comparable area for both arms of the experiment. So we see the same elongated morphology here. We also see that the cells are aligned very well in the direction of migration. They seem to be just like streaming across the callosum over to the other side. But in the knockout cells, we see that the cells are actually completely disorganized and in some cases even crossing each other's paths. As we discussed in the introduction, plexins interact with semaphorins. That's how they how their function has been understood for decades. They interact in tandem with each other. But there's actually growing evidence that plexins act independently as mechanoreceptors. So essentially, the idea is that they stick out of the cell and they kind of have this locked and unlocked formation. And what happens is that they're capable of being bent when they make contact with the environment, and this allows this to transduce mechanical information to the cell. So it gives it an idea of the cell, of where they are, what they're surrounded by, and how hard their surfaces are. So in this case, it could be that loss of Plexin-D1 results in loss of this mechanical sensing capability, and so the movement is just disorganized compared to normal. And then here we return to the callosum one final time. We see that the fiber tracks are running across these images. If we were to blow them up, red is tumor cells here, PU1 labels immune cells. We can appreciate that it looks like there's more immune cells associated with the tumor cells in the knockout than controls. And when we did a preliminary quantification, we indeed see that this is about a 2-to-1 ratio. There's more quantification to be done that I'll get to later. And then when I live stain these cells with a red membrane dye, and then I live image them, so these are living cells, we can appreciate that there's actually a lot more membrane shedding in the knockout cells, which is just all this debris on the right you see here. And so this led us to our current working hypothesis that the KO cells are able to extend out these long tethers, which we observed, but they're not actually able to maintain the membrane tension to pull them back. So there's a lot of shedding going on. And as a result, they're essentially leaving a trail that's immunogenic, and it's attracting these immune cells to kind of come in and go after them, as opposed to these normal cells, which are just kind of very sneaky and invade the way that GBM does. And it's very good at getting around the host immune system. So for
Jack,
a final,
you have a couple more minutes, so just pay attention
okay.
to the time.
Yeah, yes, sir. Okay, so I'll just skip past this. This is just about durotaxis, how the cells essentially lose their association for these axon fiber bundles when the plexin is knocked down. Here's one more image of that, where you see the cells associated with these axon bundles, and then in the knockout cells, they kind of lose that strong preference, and they're kind of associated with the other tissues present in the brain. So here are my conclusions. This deletion in these human-derived tumor cells seem to be associated with reduced invasion, increased tethering, disorganized migration, increased immunogenicity, and reduced durotactic preference. And here are some of the future directions that some of them are going on now. I'm also planning on returning in the fall to continue research elective. So we're hoping to put out a manuscript sometime sooner rather than later. And so with that, I'd like to acknowledge all the wonderful peers and mentors who supported me throughout this process, Dr. Zoe and Frito, my family, wonderful partner, and also the department for providing me with this wonderful opportunity. Thanks for your time and attention. And if there's any time left, I'll take questions.
Thank you, Jack. You clearly put a lot of work into this, and you made the most out of the scholarly year. So we appreciate the time you put into it, and hopefully you learned a lot working with
Thank you,
Dr. Zoe.
It was a great time.
Jenny,
We'll
do
open
you
it
have
up to
any
questions.
comments?
Sorry.
Is that me? Yeah, so Jack really did a great job. This is actually a challenging problem. I mean, he made it sound easy, but we actually were like almost doing detective work, trying to understand what Plexin-D1 is doing to the cell, to these patient-derived glioma stem cells. So, and these quantifications of very high quality immunohistochemistry stainings are really, I mean, he tried so many times, a lot of like the NCAM, all these new markers we've never tested in the lab. So, so I think he's and his presentation is also, we had a department in the neuroscience department. He did a presentation. Everybody afterwards came to talk to him, and just was truly inspired by the presentation as well as the work. So I commend for the great scholarly year that he spent in the lab.
Thanks, Dr. Zoe.
I was going to say, Jack, that to my knowledge, you are the first med student to pass through the gauntlet of Jenny's rigor and lab to this level. It's very rare that she'd even accept someone, let alone see this sort of performance. It's interesting because I think that your work is replicated in this picture here. If you look in the mirror, although there's a whole row of people sitting next to Jenny, Jenny's the only one who shows up in the mirror. She's unique, and her lab is unique. And I think that you have just demonstrated something pretty rare. So congratulations.
Thank you, sir.